Good morning, Raghuveer sir.
Good morning, Navalji.
I'm audible.
We'll start. Yeah, yeah.
Okay, sir. So let me
Okay.
introduce to you, sir. Yes. First of all, good morning to all the participant. I think today is the first expert session we are starting from now on. So it's give me great pleasure to welcome Professor Raghuveer sir. He is basically from Goa College of Pharmacy, and our department has privilege and fortunate that we have long collaboration with Professor Raghuveer in many research paper as well as uh, the computational studies. He has helped us, and we are very thankful, sir, that you agree today to deliver today's talk. Especially, this is a session for new PhD beginners,
Okay.
so I thought that your experience in computational chemistry, especially how to use the molecular modeling and computational chemistry for the research.
Hmm.
So it will be great benefit to our students. I again welcome you and thank you on behalf of the Bhaktakavi Narasimha Mehta University that you have agreed to deliver this talk. Now session is yours, sir. You can
Thank you,
take
sir.
as long as you wish, no problem. It's up to your priority. And
Yes,
thank you
sir.
very much, sir.
Thank you. So, okay. So I will share the screen. Okay, and Naval sir, I may have to quit and reopen. I will rejoin again. Okay. I think there is a
No problem, no problem, no
share
problem.
screen problem. Just connect in a minute. My screen is visible.
No, sir, we can't. Okay, 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 sir. Now it's okay.
Okay, visible. Okay, so it has echoed, so it's not an issue. Uh, okay. So, okay, so basically, we will look at the computational approaches in drug design. Okay, so I will take you to the structure based and ligand based approaches in structure based drug design. Now, before we go with this entire topic, it's a vast topic, so we will start with what is called as first, we'll try to understand the f- little bit of molecular mechanics in first 15 minutes. Okay, now molecular mechanics is such is a long big subject. Okay, but we'll try to cover up, we'll try to understand the uh, importance and its the meaning of it. So now the word molecular means related to the structure of organic molecules or macromolecules like proteins and DNAs. Okay, under proteins we look at receptors and we look at enzymes. The word mechanics basically deals with the motion, okay, or the movement of the molecules. Okay, now as we know that none of the atoms, neither the electrons, okay, nor the atoms are in a fixed position. Okay, if that would have been, then the entire world would have, entire universe or entire cosmos would have been at a standstill position. But we are in motion indicates that if we are in motion right at the atomic level or even at a subatomic level. Okay, so what is a force field? Now a force, we know what is a field. Okay, like we know what is a magnetic field, or we know what is an electric field. Okay, but here we talk about something different. Okay, when we talk about force field, we talk about the forces that are exerted by atoms on each other. Okay, and because of which the molecules land up into a motion. Okay, now this force field is coined into what is called as your an equation which is called as a force field equation. Okay, the earlier methods were known as Westheimer method or the force field method, and later it came to be known as the molecular mechanics. Okay, so basically what is the concept of molecular mechanics, or what is the concept of force field, is that the molecules are like spheres. Okay, they are like charged spheres or balls. You can imagine like table tennis balls which are connected by springs to each other. Okay, and but however, they can move around each other. Okay, they can move away, they can come close, atoms can move away, atoms can come close. Okay, and we can see different motions in that. Okay, so we talk about three different motions. Okay, that is basically this is how a force field looks. Okay, so it is basically an equation. Okay, that equation which talks about different components. I will take you through each of this component, referred to as bond, stretch, angle, bending, torsion, out of plane. Okay, and non-bonded interactions and Coulombic interactions. Okay, so the summation of all these equation, all these modules of the equation give rise to what is called as a standard force field. Okay, so we will look at each of these terms. First we come across what is called as a bond stretching. Okay, now bond stretching basically assumes that two atoms, okay, are connected to each other by a spring. Okay, now the atoms could be of different elements. Okay, or the atoms could be of different hybridizations like I have mentioned here. Okay, so what is the effect on the atom is basically its hybridization, the formal charge, and the nearest neighbors. Okay, so these three parameters basically take or influence the atomic chemical environment of an atom. Okay, so when we talk about hybridization, the sp2 carbon is different from an sp3 carbon. Okay, when we talk about a formal charge, okay, a oh oxygen is different from the carboxylate oh oxygen. That means a alcoholic oh is different. Sorry, an alcoholic oh is different from a phenolic oh, which is further different from the carboxylate or carboxylic OH. Okay, so they will have different charges. Okay, and then comes the nearest neighbors. Nearest neighbors are the atoms that are in its close vicinity or to which the atoms are bonded. Okay, so all these three parameters take influence. Then as I mentioned here, the force field parameters, okay, basically the numbers or the values basically differ from an oxygen which was part of a carboxylate oxygen. Okay, which is a hydroxyl or a carboxylic oxygen, phosphate oxygen, ester oxygen, and oxygen in a water. So each of this is understood or characterized differently. Okay, it is not just oxygen, but they are different oxygens by themselves. Okay, so we will look at each term. That is, first is called as the bond stretching. Okay, now we know in 12th standard what is Hooke's law. We have done what is Hooke's law. Okay, there is a spring and there is a weight attached to that spring. Okay, and how much that spring is stretched depends upon the weight or the force that is attached to that spring. Okay, if you attach a low weight, the spring extension is less. If you attach a larger weight, then the spring extension is more. Okay, and this extension, okay, so this weight, that is force that is applied, okay, and then the extension that is seen in the string is proportional, okay, or directly proportional, and it is equalized by what is called as a uh, equalized by what is called as a constant. This constant we refer to as a force constant. Okay, so you can look at this particular equation here. So this is the component, okay, this is the energy that is required to stretch apart two atoms, okay, or to stretch apart two atoms, that is referred to as atom A and atom B, okay, from its rest position or its equilibrium position, which is given by the symbol E, okay, and the new position is given by the symbol R. So RAB is the new distance, okay, REAB is the equilibrium distance, okay, and if you apply the Hooke's law, you get so the difference between these two distances in a squared divided by the constant will give you the energy, okay, that is corresponding to the stretching. Okay, so we'll not look into the most potential. So if I show you a video here, okay, so here you can see the bonds stretching. Okay, so they're moving apart, okay, and they're coming close. Okay, and this moving apart and coming close is characterized by this forced constant referred, okay, to the bond energy constant. Then we look at what is called as the angle bending. Now again, angle bending, I will first show you the video here. Okay, so here you can see there is an angle bending. Okay, now again the angle bending depends upon the charge on each of these atoms. Okay, now this is for example water, and there is only one angle involved in this particular molecule. Okay, this angle is basically between hydrogen atom, oxygen atom, and the second hydrogen atom. Okay, so this is again angle bending and is given by the formula again similar to that of the, or rather same as that of the Hooke's law, okay, which is given as UABC. Now here you can see ABC stands for three atoms, which forms the bond angle, which forms basically the angle, okay, and again it is directly proportional to the square of the distance, okay, and it is stabilized by the constant which is called as half KABC. Okay, so what is happens is basically the even in bond and in angle, when the structure is at an its equilibrium position, the energy is zero. Okay, the energy is zero, rather the distance is equal to zero, and hence the entire energy is zero. Let's say at equilibrium the new distance is equal to the equilibrium distance, so this particular term corresponds to a zero. Okay, and therefore all the entire value becomes zero, so the energy is zero in this case. Okay, the energy mostly changes when there is a significant difference in the distance in the between the two atoms. Now if the two atoms are very close to each other, there is a positive spike in energy. Okay, and if the atoms are again away from each other, again there is a spike in energy. Okay, and there if you look at this equation, it follows a parabolic rule. Okay, so it's a u bent curve. Okay, it's a u bent curve. So when the molecule or the two atoms are its equilibrium position or equilibrium distance from each other, okay, in such a scenario the energy is very low. Okay, so next is a dihedral motion or dihedral, or also referred to as torsion. Now this torsion is defined between four atoms. Okay, now as far as a single atom is contained or there is no molecular definitions for that, except for maybe single atom. There are no definitions. Okay, difficult to study and mostly studied using principles of quantum mechanics. Okay, whereas when you start with more than two atoms, you can start with the principle of molecular mechanics. Okay, molecular mechanics branch is much easier than the quantum mechanics. Okay, the roots however lie in quantum mechanics or experimental data. but much easier to study, okay, compared to a analysis in a quantum mechanics. Now moving on from, so if we move from bond, okay, as I told you, single atom there is no parameterization. When it comes to two atoms, the first parameterization comes is bond stretching, okay, because the two atoms can come close and move apart. Then when it comes to three atoms, then you have what is called as an angle. That means the atom number 1 and atom number 3 can come close and move apart, okay, so hence the angle. So you can see in this particular, as you could see in this picture, okay, so the hydrogens are coming, hydrogen 1 and hydrogen 2, they are coming close and they are moving apart, okay. Now when you have four atoms, okay, you define it with a torsion, okay, which is also referred to as dihedral. Now this dihedral is look, you can look at it particular image here. So the dihedral basically looks at atom number A and atom number D, okay, atom labeled as atom A and atom D, okay, how far or how close are they, okay, and this is defined by an angle that is made between two planes, okay, angle that is made between two planes. The first plane is atom A, B, and C. Okay, so they form a part of one plane here, and atom B, C, and D, they form a part of another plane. Okay, they form a part of another plane. Now, what is exactly happening here? We are looking at an angle between these two atoms. Okay, now if I just give me a minute, I'll show you one example which will give a clear understanding. Okay. Yes, my second screen is visible. Yes. Is the screen visible? Okay, just put it. Is my screen visible, Nawalji? Hello?
Yes, sir.
Huh? Okay, I've just shared another screen. Is this screen visible? I just wanted to know.
Yes, your
I want
screen is visible, sir.
is it now visible? This one? The molecular
It's
screen?
a,
Okay.
yes, it's a
So,
black screen is visible, sir.
okay, black is coming. Okay, fine. I'll have to change it.
Yes.
Okay, fine. No issues. Okay, so so what we are looking at is basically an angle here, dihedral angle. Okay, so you can look at this particular image here, this video. Okay, so you can see that there is a dihedral. This is an angle which I'm talking. Okay, the angle that is formed between atom A and atom D. Now what you can see that is atom B and atom C here are not moving. Okay, you can see again once again the video. This atom where I'm pointing right now is atom B, and another atom I'm pointing is atom C. So what is basically changing is the location of atom A here. Okay, so this angle that is formed when you view through this particular dimension, okay, is referred to as dihedral angle. Okay, so this is again referred to as a proper dihedral angle. Then if we move away, okay, so it is given by the formula which is basically u u0, which is the base energy, 1 minus cos n psi minus e psi. Okay, now I'm not discussing this much equation in detail. What basically it talks about, like when we're talking about bond and when we are talking about angle, okay, there is only two motions in that. Okay, a outward motion and an inward motion. Okay, so in bond stretching and angle bending, there was only one motion, that is moving out and coming back in. Whereas in torsion, what is happening is there is a cyclic rotation, or there is a circular motion of one atom. Now, this circular motion of one atom, okay, is defined by a cost function and the difference between the torsion, any torsion, and the equilibrium torsion. Okay, so when we have this, when we insert these numbers, okay, the phase value and the torsion angles, we get what is called as the energy for the torsional move, torsion bending. Okay, the next is the out of plane angle, okay, which is also called as the improper torsion. Okay, now what do you mean by out of plane? Okay, now if you look at certain atoms, okay, mostly the three atoms are in plane. Okay, mostly three atoms are in plane. Okay, but the fourth atom is called as out of plane. Okay, now what exactly is happening when it is out of plane? Okay, uh, there is a different angle bending. Okay, so here the angle is defined by the character Greek letter psi. Okay, here it talks about the bending of this particular atom, okay, either in the plane or going off the plane, this particular atom D. Okay, you have seen the difference here. Okay, so what you see here is A, B, C are in plane. Okay, but D is connected to C. Okay, so therefore it gets a zigzag structure, and the angle is between the zigzag. Okay, whereas in out of torsion, you see that the fourth atom D is not connected to C, but it is connected to B. Okay, so this angle are we talking, the angle which is made by the atom D to the plane which is made by the rest of the three atoms. Okay, and formula for that is given by this, where U stands for the energy, okay, K stands for the constant, sine function, and the psi value. Okay, the psi value is basically the angle that is made between the uh, plane and the atom that is out of plane. Okay, the next part is basically referred to as bonded non-bonded interactions. So what we saw up till now, that is atom 1, A, B, C, and D, they are connected to each other. Okay, and they are referred to as the bonded elements, okay, or bonded interactions. That means atom 1 is interacting with B, atom 1 or atom A is interacting with atom B, atom A is interacting with atom C by relationship of bond angle or angle bending, and atom A is interacting with atom T by a dihedral or a torsion. Okay, now here we look about atoms that are not connected to each other and normally lie after the fifth atom. So that means interaction between the first atom and the fifth atom, or interaction between the first atom and the sixth atom. Okay, so technically they are, though they are bonded by three or four atoms between them, but the interaction is totally through space. Okay, the interactions through space, that is, there is no direct covalent bonding involved. Such interactions are referred to as non-bonded interactions. The first one being the van der Waal interaction, okay, which is given by an equation which is called as a Lennard-Jones potential 1216. Okay, you can see the equation ULJ, that is the energy because of the Lennard-Jones potential, is given by C12 R12, okay, minus C6 R6. Okay, now here basically they are the constant. C12 and C6 are the 12th and the 6th constant with regards to repulsion and attraction. Okay, and R is basically the distance, okay, during repulsion and during. Okay, now the hydrogen bond also is a non-bonded interaction. and is given by a different softened version of the Lennard-Jones potential, which is given as 12.6. Instead of 12.6, we use a 12.10 potential. Okay, so that is for the curve smoothing or the potential smoothing. Now, if there are any two atoms which are charged, okay, in the system, then the energy between that can be calculated based on the Coulombic interaction, okay, which is simply given as, which has been learnt in the lower classes, okay, as 1 by 4 pi epsilon 0, which is basically the dielectric constant of the medium in which the charges are being placed. Q1 and Q2 are the charges on those atoms, and RAB is basically the distance between the two atoms. Okay, so we can find out the energy between these two atoms, okay, energy of interaction between these two atoms by the Coulombic equation, which is given as on the screen. Okay, so these totally together, when all these terms come together, okay, we get what is called as the force field equation, which can be seen in this particular page. Okay, so when we summarize all these, okay, this is done for all the atoms. Now let's say, just give me an example here. Okay, let's say there are four atoms. Okay, let's say there are four atoms. Now in these four atoms which are connected, these four atoms which are connected, okay, the any two atoms, okay, which are bonded, like up till fourth position, okay, or all the atoms which are bonded by a 1-2 relationship, okay, will be evaluated for bond stretching, will be evaluated for bond stretching and will be calculated, and the energy for that will be calculated using the first. So if there are, let's say, four atoms, okay, which are connected to each other in linearity, okay, A connected to B, connected to C, and connected to D, in that case, bond AB, okay, energy of bond AB plus energy of bond BC and energy of bond CD. Okay, so three bonds will be connected through energy bonds. Okay, hence the energy, bonded energy, will be calculated. Now, when it comes to angle, you will see that the angle is between four atoms. Okay, no, angle between them is four atoms. So ABC will be one angle and BCD will be the second angle. So you will have only two angles in this particular scenario. Okay, so moving ahead to the torsion. So torsion is governed by connectivity between four atoms. Okay, so first A, B, C, D. Okay, there will be only one torsion. Okay, out of plane if there is, otherwise the term will go to set to 0. Atoms after fourth position, okay, non-bonded will be calculated, and okay, and again if the two atoms are charged, interaction between two atoms will be calculated by the coulombs. Okay, so that is a brief introduction to the force field. Okay, then now let's look at the molecular model, first part, that is molecular docking. Okay, I will explain the molecular docking and then take you through a tutorial as to how as to carry out simple docking. Okay, so what is basically molecular docking? Okay, now why I have taught this force field is because without force field, none of these softwares, whether it is docking, whether it is pharmacophore modeling, screening, dynamics, okay, none of these models work, these modules work if there is no force field. Okay, so if you cannot calculate the intermolecular atomic energy, okay, then the which is calculated mostly because of the force field or intermolecular energy, then all these softwares will be basically laid to waste. Okay, there is none of these softwares work without a baseline, the basic code, that is force field. Okay, so what is molecular docking? Okay, molecular docking is basically just like how a boat or a big ship docks into a harbor. Okay, we refer to that as a docking. Okay, so in this case, okay, molecular docking, one molecule, which is basically a small molecule, okay, which is the ligand or referred to as a drug, okay, or an inhibitor, basically docks into the a cavity, okay, that is created or that is built up in a protein. Okay, this protein could be either enzyme, or this protein could be either um, a receptor. Okay, now basically there are docking involves two parts. Okay, one is the prediction of the ligand conformation, and second is the orientation or the pose in the binding site. Okay, so there are two aims of docking. First is to finding out the position, and second is the scoring of this position. Okay, now binding site is basically a cavity inside the protein, whether it is an enzyme or a receptor. Okay, it can be correlated to the cavity inside a lock. Okay, the cavity inside this binding site in the protein can be correlated to a cavity inside the lock in which the key is inserted. Okay, so when the ligand binds to this, okay, either the receptor is supposed to perform a function, or if a ligand binds to the enzyme, it is supposed to prevent the function. Okay, so either it is an agonist or an antagonist or an inhibitor. Okay, so when it comes reference to the receptor, we refer to them as agonist and antagonist, one which will facilitate the action or one which will block the action. Now when it comes to the enzyme normally we have inhibitors. Okay, now enzymes are biocatalyst, and they are like chemical catalyst. They increase the decrease the runtime or increase the production of a substrate being getting converted to a product. So now if it is interfered, if the enzyme binds to a inhibitor or a ligand and the action is blocked, so no substrate is getting converted to the product. Okay, so there is the substrate product conversion is stopped, and by doing so a therapeutic effect is elicited. Okay, we get a therapeutic effect by this particular concept. Okay, now there are multiple theories. Okay, one is called as the lock and key theory, which is called proposed by Fisher. Okay, so it believes that the inhibitor, the ligand, or the agonist or antagonist, these are like keys which can fit into the keyhole. Okay, and either by moving or not moving, they exert the action. Okay, the second is a Koshland theory, in which is referred to as an induced fit. Now, induced fit means where as the ligand binds to the protein will change its conformation or its shape. Okay, and by doing so, it will produce a therapeutic or whatever activity has to be elicited. Okay, so how it is done? When the ligand slowly binds to the protein slowly undergoes a deformation. Okay, and okay, this protein slowly undergoes a deformation change. Okay, thereby exerting its action, either therapeutic or non-therapeutic. Okay, so now, how the sampling, and there are multiple ways in which sampling is performed. Okay, one of the simplest is genetic algorithms. Now, in genetic algorithms, what is done is the okay, now in genetic algorithms basically obeys the principles of Darwin. Okay, which indicates that as the generation changes from one to another, some changes are incorporated. Okay, and there is a evolution. Okay, there is a Darwin's evolution. So it is also referred to sometimes evolutionary algorithms. Okay, now basically the conformation and the position, as I told you, there's a conformation, okay, of the ligand, and there's a position of the ligand in the binding site. Okay, these can be stitched, or this can be connected to the connected to a genetic algorithm. Okay, so whenever the change is performed, whenever there is change performed, so either in the conformation or either in the position, it is retained or rejected based on the interaction energies. Okay, there is a retention or there is a discard. Okay, now retention is done when the energy scores are better. Discard is performed when the energy scores are not better compared to the current position. Okay, so if there is a current, if there is a energy calculated for a current position, and then there is a change introduced, Now, if this change introduced has got better energy profile, then this change is retained, otherwise it is rejected. Okay. Now, what is how it is accepted or rejected? So there are two algorithms, as I told you. One is the search algorithm, basically which search for a comfortable position, okay, of the ligand in the binding site, okay, either by moving it or twisting and turning it. Okay. Now every twist and turn or every movement, okay, has to be scored. It has to be ranked. Okay. This is done by called as approach called as scoring function. Okay. A scoring function here means an equation. Okay. So what it does is, when the atoms of the protein are in close vicinity with the atoms of the ligand, okay, just like the force field I have shown you earlier, okay, based on that force field, interatomic distances are calculated between the protein and the ligand, okay, and it is converted to some feasible number, a number which represents the goodness of binding, or okay. So there are basically three force fields. One is called as the three scoring functions. One is referred to as the force field based function. Second is referred to as a empirical scoring function, and third is referred to as a knowledge based. Okay. Now in this particular, as I told you, there are Lennard Jones potential. Okay, and there is Coulombic forces of interaction. These are the non-bonded interactions which are basically seen. Okay, so when you have to compute between the protein and the ligand, you will compute these three. Okay, you can also compute a hydrogen bond interaction. So you will calculate hydrophobic interactions and hydroelectrostatic interactions. Okay, and within van der Waals, you could either have the hydrophobic interactions, or you could, let's just, okay, or you could have what is the hydrogen bond interactions. Okay, I will show you then. There are a lot of two scoring functions available. Okay, like dog score, auto dog score, gold score, Rudy. So different functions, X score. Okay, but normally we look at auto dogs if you are using the simple tool. Then coming to the docking methodologies. Okay, docking takes place in three principles. Okay, where one is considered when the ligand is rigid and the receptor is rigid. Okay, now what do you mean by rigid? Means the atoms are not allowed to move. Okay, that means the bonds and the angles are kept constants. Okay, and as far as the receptor is concerned, even the torsions are kept constant. Then in case of the ligand, okay, ligand is also considered rigid. That means it is fixed in one conformation, and multiple such different conformations are generated. Okay, the conformations for the ligand are not searched in the binding site. Okay, so different conformations are done. Okay, and each conformation is moved. There are three translational and rotational motions. Okay, what is a translational motion? Okay, now translational motion is moving from one location to another, okay, either in the x direction, y direction, or the z direction. I repeat, a translational motion is you are translating. Okay, that means moving an object or an atom from one place to another in three directions. Okay, and the second motion is called as a rotational. That means the body or the atom rotates or tumbles, okay, at its mean position. It is not moving anyway, but it is just rolling. Okay, now this rolling can take place either in one x direction, it can take place in y direction, or it can take place in z direction. Okay, so this way, if we look at it, we have got 6 degrees of freedom here. Okay, so you can move and then rotate. Okay, you can move and then rotate, you can move and then rotate. Okay, so you can keep on doing this move and rotate or translate and rotate functions. Okay, till you find out a ideal position. Now, whenever you do translate and rotate, every translational and rotation is scored. If it is good, it is accepted. If it is bad, it is rejected. Okay. So let me take you to. Okay. So brief stages of your drug discovery basically involve target identification. That means you need to know the target. What is the target? Target here is basically a protein, okay, which whose function causes either a whose function is involved in the pathophysiology, okay, or the disease condition. Like if I give you an example, you have ACE inhibitors, okay. You have got acetylcholine esterase enzyme, which is responsible for Alzheimer's disease. Okay, so these are called as targets or validated targets. Okay, now hit identification means to find out some molecules quickly, okay, which produce some elicit some action, desirable action. So hits are normally in few numbers, okay, followed by lead. Okay, a lead is a molecule which is the best amongst the hit and which is subjected to further modifications in the chemical structure so that the biological activity and the pharmacokinetics may be improved. Okay, and the rest follows are the preclinicals, that is animal testing, human testing, approval by the FDA, and subsequently moving into the market. Okay, so based on computational chemistry studies, many molecules are available in the market. Okay, and advantage of computational chemistry, okay, it basically has significantly reduced the time and the cost associated with finding and optimizing lead compounds. Okay, so okay, in normally in traditional methods what happens is you first identify new therapeutic agents using experimental and rely heavily upon the biological system and the natural sources. This process is normally time consuming, it takes lot of years. Okay, just if I give you an example, it will take 3 to 4 years just to get a molecule to clinical trials, okay, from a traditional drug discovery procedure. Whereas the rational drug discovery, which basically employs the computational tools, this process can be triggered faster. Okay, that means within a period of 6 to 9 months, you can come to clinical trial phase. So you can understand the depreciation fall in the timeline. Okay, so from 3 years, you'd come to 9 months. Okay, or rather 3 years, which is like 36 months to 9 months, which is 4 times the reduction in time space. Okay, so in the 4 times the reduction in time space, so that's the advantage. Okay, so for optimizing, you can see many molecules here which I'm showing. Okay, like sakinavir, which is an HIV protease inhibitor. Okay, then you have zanamavir, which is a neuraminidase inhibitor. You can see the structures and all identified using structure based or QSAR methods. Okay, and validated using different tools like molecular dynamic simulation, and before moving in actually into synthesis. Here you can see the baloxavir. Okay, you can see the tivozanib. Okay, these are VGFR inhibitors. Okay, and anti-influenza inhibitors. You have imatinib, which is a cancer treatment. Okay, it's a tyrosine kinase inhibitor. You have dorzolamide, which is used in a treatment of glaucoma. It is a carbonic anhydrase inhibitor. These all molecules are identified using computational chemistry. Okay, raltegravir, okay, which is a virus inhibitor. Then capmatinib, which is basically another molecule used in the treatment of cancer. And ritonavir, which is used in the treatment of HIV. Sitagliptin, okay, which is used in case of the diabetes treatment. The dose given is smaller compared to that of your. All these molecules have been identified through a rigorous computational chemistry process. Okay, so we have looked at this, we have looked at the equation. Okay, so we'll move molecular docking, which is basically part of structure based drug design. Okay, So what is this is how we are searching. Okay, so what we are looking here is, so you can see the blue structure. The blue structure is basically the uh, reference structure, and the docking identifier. So you can see different conformation and different positions the ligand takes place in the binding site. Okay, so here you can see that the protein is constant, it's not moving. What is moving is the ligand. Then, as I told you, there are three aspects, which is rigid docking, semi-flexible, and flexible docking. Now, in rigid docking, you have got protein and ligand both held rigid, okay, and the position of the ligand in the binding site is found either by moving around the binding site or rotating the molecule in the binding site. So, advantage is speed. So, you can see this is the fastest docking procedure, okay, and it can be used to screen abundant chemical libraries or million to billion chemical libraries in a top-notch speed. Okay. The other side you see, which is called as a flexible docking, which is highly accurate because the entire binding site of the protein is set flexible and the ligand is also flexible. Okay. So there is a squaring. Okay. So flexibility has squared up here. So flexibility squaring up meanings changes that are to be made to the protein and ligand are abundant. Okay, and because of this you can get better solutions, so the accuracy is high, but however the time required, the speed is slow, because there are a lot of changes to be done. Like as in case of rigid docking, what you see that the protein and the ligand are rigid. Okay, so the only motion is allowed is translational motion, that is moving up, down, okay, left, right, forward, backward, and tumbling during all those sessions. Okay, the next one is a semi-flexible docking, which is like intermediate in accuracy and speed. Okay, most of the softwares use the semi-flexible docking approach, wherein the just give me a minute. Okay, so semi-flexible is one of the appreciated approach, wherein the protein is rigid, whereas the ligand is allowed to flex. Okay, so the speed and the accuracy is intermittent between the rigid and the full flexible docking. So ligand, in addition to the translational and the rotational motion of the ligand is also allowed to flex. That means it is also allowed to change its conformation or change its structure. Okay, so therefore the speed decreases but accuracy increases. Okay, so then how to go about molecular docking is, first is protein preparation. Okay, now protein preparation involves the identification of a target protein, enzyme, okay, or a receptor which is of therapeutic interest. Okay, which is of therapeutic interest. Okay, and then the crystal structure is obtained from what is called as the protein database, or nowadays you must have heard a lot of AlphaFold. Okay, which is basically three-dimensional structures of protein which are predicted. Okay, now protein database is basically a crystal structure bank. That means proteins are crystallized. Okay, and proteins are crystallized, okay, and available to study in the 3D computer world. Okay, now when we talk about protein preparation, okay, there are two parts to it. One is called as a removal of the non-essential structures. Now when protein is prepared, when protein is crystallized, okay, there are a lot of um, other molecules that are added to stabilize the protein movement and bring about a perfect crystallization. Okay, uh, so we remove all these non-essential entities like waters, okay, or other ligands that been added, okay, and clean up the protein. Second, when crystal structures of proteins are being performed, okay, the hydrogens are missing. Okay, the hydrogens are vibrating so fast that the capture of the hydrogens under X-ray motion or X-ray beams is of quite some difficulty. Okay, so in such cases, only heavy atoms like carbon, nitrogen, oxygen, phosphorus, okay, sulfur, these atoms, their locations are detected and converted into the crystal structure. Okay, further we have to assign what is called as the charges, okay, which could be formal and partial charges to all the atoms in the protein structure. Okay, this is because if the atoms are not charged, okay, then you will not able to calculate the interaction energy. Okay, as we know that interaction energy that we calculate is between non-bonded atoms. Okay, either van der Waals hydrogen bond, okay, or the electrostatics. So if there is no charge, okay, then your molecules will give for fictitious results. Okay, then binding site detection. Second, binding site is, as I told you, is the site in the protein where the transformation normally takes place. So when you design an inhibitor, the inhibitor is designed to block that transformation, prevent that transformation from happening. Like if I take the case of acetylcholinesterase, okay, this is an enzyme which transforms or degrades acetylcholine, which is a neurotransmitter, to acetic acid and choline. Okay, so if now by doing this, the neurotransmitter acetylcholine, the concentration drops, okay, and that results in Alzheimer's symptoms. Okay, so to avoid that, the steady concentration of acetylcholine needs to be kept in the brain, and that is by doing inhibiting the acetylcholine or the butyrylcholine esterase, cholinesterase inhibitors. Okay, these cholinesterase inhibitors block the site, okay, and prevent the transformation. Okay, so here you can see this is the binding site, okay, of the protein where the ligand, okay, if I'm right, that is, okay, that is in the cavity. Okay, you can see the surrounded are the amino acids which exhibit some interactions. Okay, so this way we can detect the binding site. Okay, now let's look at the, okay, so like this is the drug, let's say this is a protein which is referred here to as DPP4. Okay, now what we normally do is we have a structure, okay, we have a structure, we can draw the structure, okay, or we can create the smile. Okay, so for this I will go to the website. Okay, I will take you to the website. This website is okay, so this is to draw the structures. Okay, so this is the website where you can draw the structures. Okay, so here you can draw a structure or you can provide a smile. Okay, you can draw the structure here. Okay, these structures, let's say if I draw the structure of a simple paracetamol. So I draw the structure of paracetamol. You can see that immediately there is some mol structure being created. So from this I can move on to drawing a structure with regards to Autodock, which is a free software for docking available. I can generate the three-dimensional coordinates for this structure. I can add hydrogens and I can adjust the pH for a docking. Okay, so with this. So you can see here, okay, the structure of paracetamol has been created. However, the software misses to add the charges. Okay, so this way you can download the structure and use it further for drawing. Okay. So scoring function I've already discussed. Okay, the scoring function basically is responsible for giving a number. Okay, which will tell you whether the docking has successfully produced or not. Then we first what we do is we call as docking validation. Now, docking validation is a procedure to ensure the methodology and scoring function produce accurate and reliable results. Okay, with which we can perform further docking of our synthesized molecules or designed molecules. Okay, now the basically the docking validation procedure involves, okay, so that is the objective. What it involves is docking of the crystal ligand. Now here you can see the crystal ligand which is in rust color. Okay, the rust color here. So you can see structures in uh, teal color. This is teal color. You can see in magenta, in cyan color. This one. These are all cyan atoms, and you can see in rust color. Okay, so what the docking procedure should do is basically. Uh, Validate. Okay, so you can see what will happen here. Okay, so this is the docking taking place. Okay, and it is searching for different positions in the binding site. So you can see it is rotating and translocating in the binding site. Okay, so these are the different steps that I have captured. Okay, this is not visible during the docking per se, but I've just captured it. Okay, and you can see that eventually docking has taken place. Okay, with some RMSD here, with some small error on deviation, but you can see that the majority of the cyan atoms, okay, oxygen atoms, nitrogen atoms has overlapped. So the docking position, you can see there is a clear overlap, okay, between the docked position and the crystal position. Now when this is achieved, you can say that the procedure of validation is complete. Okay, so as to ensure that the methodology and function do produce accurate results. Okay, now let me give you an example how to carry out docking, simple docking with your structures. Okay, just to get a feel whether your molecule is going to bind or not. So as I told you, you need to know which protein is that you are targeting. Just give me a minute. So we will go to a website called as SwissDoc. Okay, so in this Swiss dock, you will select the option which is called as docking with Autodock Vina. Okay, now here they have given an example that you can try out with. So here the protein is basically beta lactamase. Now all we know that beta lactamase is a protein that breaks down antibiotics like penicillins and cephalosporins. There is a beta lactam ring in penicillins and cephalosporins, which is when intact exhibits the antibiotic action. But if the ring is opened, okay, its antibiotic action is destroyed. Okay, so now this particular, you can see there is a PDB, which is protein database or data bank ID, and given code is okay 3G31. Okay, so the PDB code is 3G31, which is, if you download this crystal structure, it is basically a beta lactamase protein which is bound to a cyclic urea. Okay, you can see different crystal structures here and different proteins like dabrafenib bound to BRAF protein. Okay, so we'll look at this, how it works. Okay, so first option here is to provide the smiles. Okay, so first choice is you have to do what is called as choose the docking with Autodoc Vina. Now, once you do that, first option is to submit a ligand. Okay, now to submit a ligand, you can submit a smile of a structure. Okay, you can draw and obtain the smile. Like, for example, okay, now I have drawn the structure in MDN and I want a smile of this. Okay, so here you can see that a smile has been created for paracetamol. Similarly, the compounds that you synthesize or the compounds that you design, you can draw here. Okay, run it as a input format as a mol. Okay, output format as smile. Now, smile does not have 3D, and smile you cannot add hydrogens. Okay, again you cannot set the pH. Okay, so once you have the structure, you create the smile, you come back to your website, that is doc position. So you can submit a file here either in smile format, or you can draw the structure directly, or you can upload a mol format or a pdbqt format, or you can draw the structure. If I'm there, so there is a sketcher. If you click on the sketcher, okay, you can upload a PDB structure. Then next is to submit the protein, okay, the target. Now this target, either you can submit it, if you have the PDB code, you can submit the PDB code here and it will accept the protein. Okay, if you do not have the PDB code, then you can upload the protein from this choose file browser. You can browse the structure from your browser option and then upload the structure. Okay, and then you say prepare the target protein. Okay, once the protein is done, okay, target, once the protein is loaded, you need to set the binding site. You need to set the binding site where the ligand is going to bind. Okay, once the binding site is approved, you need to look at this search box, center box, okay, parameters. Now normally the box size that we keep is around 20 angstroms cube. Okay, that means it is 20 angstroms in the x direction, y direction, and 20 angstroms in the z direction. And the box center we can do by clicking the ligand, which I will show it to you. Okay, so this will define the binding space. Okay, so this is where the ligand will bind. So next you need to check the parameters, okay, like how many. Then you can set in the email, okay, give a name for the docking, and the docking calculation starts. You can either save this bookmark, okay, or you can wait. The docking normally takes place in 2 to 3 minutes. So here you can see that the summary of the query is given. Okay, like what is the ligand structure, what is the name of the target, what is the method used. Okay, then uh, what is the parameter boxes, everything. Okay, and which papers you need to cite when you use this particular work. Okay, then you can see the ligand that is given for your use. Then you can see from here the results. Okay, you can see the binding results. Now you can export the results as a zip file from here. You can see there is an export option. Okay, so this export option will allow you to download all the results and all these numbers. Okay, all the docking scores, all the docked poses will be available for further study in your now, how to do next thing is once you have the ligands with you, you need to do what is called as the binding site analysis, like how to find out whether your ligand is binding. Now, one thing I have not discussed in this thing is the proteins or the peptides. Okay. Now, it is very mandatory for you to know the structures of the amino acids. Okay. And what type of interactions these amino acids exhibit. Okay. Because without which you will not able to detect the interaction between the amino acid and your ligand. Okay, so that is a important or a critical step. Okay, so know your amino acids. Okay, know their single letter codes, know their three letter codes. Okay, so if you look at this particular image here, बंद कर, बंद कर. Okay, so is this image what you can see basically is the backdrop you can see which is lightened in color. It is basically the protein itself. Okay, it is not a background for the image. Okay, it is the protein itself. Okay, you can see the amino acids which are in teal color. Okay, the red tips of the steel color are basically oxygen atoms, and the blue tips on this are basically the nitrogen atom. So here you can see alanine, okay, which is given by ALA code, and the number is 898. That means it is the position is 898 in the protein structure. Okay, so this is the structure. There is glutamate, okay, which is a glutamic acid, okay, which has got again structures here. Okay, then you have got tyrosine, okay, which is an aromatic acid, and simultaneously it is polar. Then you have another tyrosine here. Okay, so you can see aspartate here. Okay, aspartate is a side chain is uh, side chain is again acidic. Glutamate side chain is acidic. Okay, then glycine is just the backbone. There is no side chain to glycine is a very simple amino acid. Okay, and you can see here how to do binding site analysis. So this way, what are the interactions that you need to figure out, whether there are hydrophobic interactions, hydrogen bond interactions, pi stacking, cation pi stacking, and halogen bonds. Okay, so these are normally interactions which we scout for when we do a binding site analysis. Now, in this particular image, you can see that there are gray dotted lines. These gray dotted lines indicate the hydrophobic interactions. Okay, then you can see there are blue dotted lines. Okay, the blue dotted lines basically indicate there are hydrogen bond interactions. Then you can see small tennis ball sort of things here, which is at the center of the rings. Okay, one is in the center of the ligand ring, one is in the center of the tyrosine ring. Okay, and then you can see there is a green dotted line. Okay, this is referred to as a pi stacking. Okay, there is a since these are aromatic structures, since these are aromatic structures, okay, they have pi clouds or pi electrons. Okay, and one cloud which is electron deficient can stack with another cloud which is electron rich. Okay, now this particular structure does not have any halogen atom, otherwise we would have there would have been a chance to see a halogen bond also. Okay, so here you can see, okay, the interactions. I have put it on a cradle view. Okay, so here you can see the interactions between the, so it's, you can see that the structure is in a cradle view. Okay, it is moving left and right slowly. Okay, you can, I have put it on again, you can see there is a motion. Okay, so you can see the whole protein is moving. So you can see now that background is not the background, but it is actually the structure of the protein. Okay, and you can see that there is a, you can even see the pie stacking, you can see the gray lines, you can see the green dotted line between the two ping pong balls in the center of the ring. Okay, so this way you need to analyze what are the interactions, okay, so that you can do a further optimization in your ligands. Okay, another crucial part in molecular modeling or when you do drug discovery is ADMET. Now ADMET is basically absorption, distribution, metabolism, and the toxicity of a structure. Okay, now this particular screen, you can see there is, we will look at what is, how to find out ADME parameters. Okay, now you can see a structure has been sketched. Okay, and or you can load a smile of the structure, either way. So either you draw it or you load it. Okay, and then you have to run the calculation. So when you run the calculation, you will see that it goes in and processes the properties for you. Okay, it shows in properties for you. Okay, here you can see the different properties. Okay, like physicochemical properties. Okay, number of hydrogen bonds, what is the molecular weight, what is the total polar surface area, number of hydrogen bond acceptors, number of hydrogen bond donors. Okay, how much or how many rotatable bonds, you can see all in this particular, then log p calculated. Now you can see that this particular structure here, okay, this is this pink, this is called as a radial plot. This is called as a radial plot. Okay, and in this radial plot you have got different properties, okay, like polarity, insolubility, so flexibility, lipophilicity, and you can see that this particular molecule is within the range. Okay, is within the range. It's all red lines basically indicates the properties of the molecule, and the red or the pink space or the pink web, a pink hexagon basically represents the ideal properties. Okay, so you can see that this particular structure has got ideal properties because it is in, lying in the pink zone. Okay, so further on you can look at other properties like absorption, whether it will show GI absorption, whether the molecule will have a blood brain barrier permeability, then whether this molecule become a substrate for hydrolysis or breakdown using the cytochrome P450. Now cytochrome P450 are enzymes that are present in the liver. Okay, so anytime whenever either we have a xenobiotic, that is a ligand or a chemical molecule that we consume, okay, or which is a part of food, or which is xenobiotic means non body parts or non body structures. Okay, so small molecules which not do not originate because of the body but taken from external source. Okay, so such molecules will undergo metabolism. So metabolism is basically breakdown or transformation of the chemical structure so that it is easily excreted from the body. Okay, and then we have excretion parameters. So if this particular profile for the structures that you synthesize or design before synthesis, it is ideal you check your structures whether it lies, whether the design structure has got good ADME profile, then I recommend you proceed for synthesis. Okay, or else if you see that the profile is bad, you do some changes to your chemical structures with which you feel that the biology could be improved. Okay, so this way you can carry out the ADME properties for your molecule. The next is Property Explorer, which is an open molecules software. These are all open tools. There is no, there is no financial burden, okay, either at the institute level or at the researcher level. These all tools are available free online. You need not even install them on your computer, okay. So this is another one which is called as a Property Explorer. Now this Property Explorer, again, you have got a drawing module, so you can draw the structure of your compound, okay, and it will tell you some basic toxicological properties, okay. It will talk about the basic toxicological properties. So if you, once you draw the structure and click on it, you will see toxicity risks and everything on the right hand side column. So this is a structure of aspirin. What you see that it is showing little bit of mutagenic and teratogenic properties, okay, and effect on the reproductivity. Then its log P, you can see, is green. Solubility, molecular weight, and total polysurface area are green. Okay, drug likeness is better, but drug score is poor. So red indicates bad, yellow indicates caution, and green indicates safe. So you can see these properties. Okay, now these are all predictions, not actual. Okay, so you can see for other structures. Okay, so this is aspirin. Okay, now this is race ibuprofen. You can see this is all green. Okay, in ibuprofen you can see this is all green. This is your ibuprofen, so it is a safe structure. Okay, then you can see the next structure, which is celecoxib, which is again another safe structure, though its drug likeliness is less, but it's a safe structure. It has been used, available in the market, but however its predecessors like rofecoxib, valdecoxib, they have been withdrawn from the market because they cause a threat to the heart. Okay, they can cause non-rhythmic or arrhythmic beats. Okay, the heart has a rhythm. Okay, it beats in a rhythmic pattern. Okay, so the drugs which bind to COX inhibitors, okay, they can destroy that rhythm of the heart. Hence, they are called as arrhythmic in nature. Okay, causing arrhythmia. So this was basically about structure-based drug design. We look at a new other part, that is ligand-based drug design. Okay. Now many a times there is a possibility that we do not have the structure, the protein structure to carry out docking. Okay. Then the we have to heavily rely on the molecules that have been explored. Okay, with similar biological activity. So what we could do is we can do what is called as quantitative SAR or QSAR. Okay. So in quantitative QSAR, what you could do is, if you have structures, now this is only possible if there is a previous knowledge available. Okay. If there is no previous knowledge, quantitative structure activity relation cannot be performed. So you need previous knowledge of structures which have biological activity that is of your interest. Okay. As far as the descriptors or chemical characterization or descriptors, as we call them, okay, or properties that describe the structures, can be calculated using tools. Okay, now you can subject these structures and the activity to a machine learning algorithm. Okay, and it will give you what is called as a predicted model. Okay, predicted model is basically an equation which will help you to predict the activity of your newly designed compounds. Okay, then next we look at what is called as a pharmacophore. Okay, now pharmacophore again is an ensemble of steric and electronic features of the molecule which is required for optimal interaction with the biological target. Okay, even if you do not know the biological target, but these are the features that are required and found and lead to desired biological activity. Now, which are these different features? Okay, these different features also are seen when you do docking. Okay, you will see a hydrogen bond donor, you will see a hydrogen bond acceptor, you will see an anionic feature, okay, you will see a cationic feature, you will see an hydrophobic feature, and you will see a aromatic feature. Okay, so these are the features that are required. Not all, they are dependent on the binding site, okay, which we or we may not be aware of. Okay, so based on these features we can do what is called as, so if we have to identify these features in the molecules of our interest, okay, then next we do what is called as a similarity search. Okay, similarity search again is the website directly. If you click on similaritysearch.ch, okay, you will land up. So these are different tools under the same Swiss drug design. Okay, so we looked at Swiss doc. Okay, we looked at Swiss ADME. We will look at what is called as Swiss similarity. Okay, now again in this case you start off with a known molecule, a molecule whose activity has been performed. Okay, now this is not a part of a de novo drug design. This is not a part of a de novo drug design where you're trying to find out a new drug all from its right from scratch. Okay, but you have prior knowledge about some molecules that already exhibit activity, and or there are innovator molecules. Okay, so innovator molecules are the first molecules in line which have been released in the market, okay, and are safe to use. Okay, so if you know some innovator molecules, you can start off with the molecule and try to find out similar molecules from your database, different databases. You can either provide a smile of the structure of the innovator molecule or a molecule of active known activity, or you can use the sketcher to draw the structure. Okay, let's say we start from any of these molecules, like you take propranolol, you take diclofenac. Okay, you can start with the molecules of this nature. Okay, so similarly you can activate the sketch machine here. So once you activate the sketch, you can draw the structure if you do not know the smile. Okay, if you know the smile, you can copy paste the smile, or you can know, if you know the structure, you can draw the structure. Once the structure is drawn, okay, you move into what is called as finding the database. Okay, now there are chemical libraries attached to this particular server. Okay, based on its synthesability or based on its direct purchase availability. Okay, or the drug bank itself, the molecules which are already reported as drug. Okay, now here you can, there are different libraries available from different vendors. You have Enamine, you have E-molecule, you have got Life Chemicals. Okay, so there are different chemical databases based on the choice made here. Okay, based on the choice made under the select a compound class. Okay, these libraries here can change, remain same, or can change. Okay, now these terms here which you can see, that is FP2, EC, FP4. Okay, these are basically referred to as descriptors or search patterns. Okay, so FP2 stands for fingerprint 2, EC4, ECFP4 stands for the extended connectivity fingerprint 4. Okay, which is a better descriptor compared to FP2. Okay, then you can use a generic scaffold. That means if you want to just use let's say your drug contained a benzimidazole molecule, and you click on this generic scaffold and click on any of these items here. Okay, so this database will start screening, this module will start screening the database and find out all the possible benzimidazoles available in the library and throw it up front. Okay, so it will, once it starts screening. Okay, so here it will show you what are the job parameters, what is the query that you have submitted, what is the library that you are screening, what is the method of screening. Okay, and estimated of time of submission, and exact time of submission, and estimated time of completion. Okay, so once this done, this it shows you what is your query. Okay, so here you can see it was benzopyrazole which was taken structure. Okay, and you can see that it has created similar structures. Okay, based on similarity. Now you can choose these structures and you can submit them to docking if you know they are uh, Okay, so here you can see I've chalked out a virtual screen flow. Okay, this virtual screen flow helps you to um, understand the whole scheme that you need to do. Okay, now virtual screening is basically if you are not going to like if you have not designed any compounds, but if you have a primary target, okay, of biological target, you can run what is called as a virtual screening. Okay, virtual screening is basically finding like a needle from a haystack. Okay, or rather many needles in a haystack. Okay, and these needles can be further optimized for their size and dimension, something like that. Now the molecules that you obtain from virtual screening, okay, you can if you have molecules that are already known, then you can prepare a QSR, okay, model, and from the known inhibitors, and then you can use these known inhibitors post validation to find out the virtual hits, okay, and predict their activity. Okay, then you can perform the ADME toxicity studies, and you can weed out the molecules that are not properties who are deviant type. Okay, whenever again virtual screening, what you could do is you can weed out whose docking scores are not good. Okay, again in case of QSR, you can remove the structures after prediction whose predicted scores are not good. So you have a 3-point check here. Okay, so you have a QSR check, you have a virtual screen check, and you have got a ADME tox filter check. Once these checks are performed, so from millions of molecules, you can boil down to some few hundreds or few thousands molecules, which you can use for study. Now, there are two approaches while you want to study this. Either you can buy these compounds directly from the vendor, okay, and perform a quick analysis, do a quick biology, do a quick analysis, and find out like from like 10 or 15 of these molecules, which molecules you think exhibit or experimentally determined to form biological activity. Once this is available with you, okay, then you can choose the molecules which are most active and then re-optimize them, which is called as the lead design or lead optimization. So you can look at what are the structural features you can modify. Okay, if you want to simplify the synthesis pattern of that structure, you can make some chemical changes. Okay, and once these designs are performed, you submit them back to the QSR software, and you submit them back to the docking software to find out whether the predictions are better or have improved or have gone down. Okay, am I clear? So you can so this way you can run a full virtual screening workflow to get some molecules. Okay, so Naval sir,
Yes, sir. Yes, sir.
so we can have some questions if any students wants to ask questions or something like that.
Right now, no, sir. It's not shown in comment box.
Okay,
Anybody have any
so
questions, students?
yeah, they um,
Any have questions?
I have not joined the talk. I think I need to join the talk.
No, sir.
Okay,
Comments is
they
already shown on my
yeah,
desktop, sir.
yeah, comments are there. Okay,
Yes.
so anyone has questions, you can have a few questions. Uh,
I think
I got
so.
I
No,
got a
sir.
comment. I got a comment to use Hindi or Gujarati. I'm sorry, I cannot use Hindi, Gujarati when I'm come. Okay, so if you can try out these tools, all the students, you can try out these tools yourself. Okay, and if you have questions, you can write to me. Okay, sir, you can share my email with them.
Sure,
Okay,
sir.
so you can, if you have questions, you can write to me. Okay, I will help you solve out. Okay, because these topics what we have covered out, I try to do it in a buffer time of 1 to 1 2 hours. Okay, but itself it's like a 30-hour coursework. Okay, we have tried to quickly fit in lot of important topics in this part. Yeah. Okay. So okay, Navaji, ठीक है?
Sure, sir. Huh.
Okay, so we can complete this session now. Yeah,
Okay.
we
Thank you,
we can
sir.
close. Yeah, thank you very
Yes, sir.
much. Yeah,
Thank
okay,
you.
take
Thank you
care.
very much,
Yeah,
sir.
thank you, sir.
To all students, kindly join with us because of we are starting a video lecture for that from the 10 minutes.
Then I got a postdoctoral fellowship as a research scientist. I was working at the Ohio State University, USA during 2009 to 2012. And then from 2012 onwards, I moved to India and I came back to the Karma Bhumi and I started my journey at the Virani Science College, Saurashtra University. And then from 2018, I'm working as an associate professor at Department of Chemistry, Dr. Kavi Narasimha Mehta University, Junagadh. So my professional experience is about 16 years after PhD. I have 42 research papers already published. I have 3 Indian patents, more than 20 invited talks at the national international level. Already 5 students are there in PhD program. Also published 2 books for the cancer-related molecule synthesis and the developments. Also, specifically, I'm coming from the chemistry background. Those students who are in the chemistry, they know that what is the name reaction, right? So if you Google my name, this is the reaction which I have discovered. It is recognized as Su novel rearrangement. Su is my professor, Professor San Long Su, with whom I was working in the Taiwan, and we together discovered a new rearrangement, and we have given the name as a Su novel rearrangement. You can just Google this name, and you will find the wonderful rearrangement from aldoxine to amines using the oxidative condition. So this is my short profile of my research. So as I told you that I'm coming from the same background where you have started your journey. I started my journey from the Kotak Science College, very famous college in Rajkot. You may be knowing, right? So I was a student from Kotak Science College, and then slowly and steadily I get the progress, and I was selected as a PhD candidate in the Taiwan, and also got the fellowship for the research scientist at Ohio State University, USA. So I just want to tell you from this slide that you will be also having a chance now, because you are going to join the PhD program. So you must target high. Okay, or you have to work hard, of course, but you must target high to get your good research profile. Okay, so that's why I'm just saying that no matter whether you are coming from any university or any colleges, right? Your work, your experience, your honesty, and your dedication is very important. So which can give you a good profile in the research. So the question always arises, why research? Is this slide visible to you, friends?
Yes, sir.
Okay, thank you. I'm just asking for every slide so that I'm sure that whatever I'm talking, you are also having the same slide. Okay. So, why research? The question is why we should do the research? Is there any benefit? Is there any special purpose? What are the
yes.
Please, somebody is have their microphone on. Please keep it muted. So, whenever you join your program, I'm telling you that when you join the PhD program, you must understand what you want to do. Why research? Is there any benefit? What is the benefit which I will get? What is the benefit to the institute? What is the benefit to the society? After all, if your research is not useful to the society or not useful to your community, that means your research is not up to the mark. So I put the 2 photographs here that's showing the contribution of some tremendous development of recently in the chemistry, especially organic chemistry. The Nobel Prize winner in 2021 and Nobel Prize winners in 2022. Whenever you see these kind of people working in the specific field, right, we must get
the eagerness to know that what kind of work they have done. Sometimes we ask the student who got the Nobel Prize in your field. When you are doing the research specifically, you must know. And students sometimes don't even know the name of the scientist. Actually, they are working in their field, organic chemistry or analytical chemistry or physical or geology or life science whatsoever. I'm just saying that you must have understanding what is your field and who are the best researcher in your field. So that is the first thing you have to understand, right? So when you do the research, what you are getting, we are adding the knowledge. We are adding the knowledge. Plus, we are doing the two important things. We are trying to solve the problem and we are trying to understand the nature, because nature has its own laws. Nature has its own reactivity. If you can understand it, you can more understand if you do the research. You can solve the problem through the research. And after all these efforts, after all, your hard work will be benefited to mankind and also benefited to nature. As you know that recently, during the corona period, you know that, right? Thousands of people were affected in India and overall, of course, right? So if people have not done research, if they have not do the research in vaccine development, what we could have done, right? So ultimately, through the research, we can benefit to the mankind and of course to the nature, right? So that's why research is very important, and you must focus on how we can add the knowledge which is existing there. Now, the question is then what is research, right? So basically, when you start your research, three things you should be having in your mind. I'm hopeful this slide is visible. The hypothesis, which is why. Second is methodology, which is how. And third one is result or outcome, which is what. So any research should have three important pillars. Number one is why. Why we want to do the research? What is the purpose? What is the objective of our research? Number two, methodology. How we are going to do this? What will be the methodology? What kind of technique we are going to use, right? So how means methodology, the experimental part. And third part is results. So you're doing some research with certain methodology, research. What kind of result you have got? What is the outcome, right? So if your research can answer all these three questions, why, how, and what, then your entire research is called the rational research. For example, every time if I ask the students, if new students are coming into the laboratory, and if you ask why you want to do the research, then always question is because my professor told me to do this, or because my family member want me to do the research. But that is not the answer of any questions. Why you want to do the research? Why means you have to find out the rationale behind your research. Then question is how to do this. Then third is what. So all these three things we are going to understand a little bit more. Okay, so let us try to understand why. What is hypothesis? Hypothesis. So hypothesis means what? So first of all, you have to identify a problem in your field. Suppose you are working in a chemistry, or if you are working in a botany or zoology. First, you identify a problem. Then, problem means what? The problem means, suppose I am talking about chemistry, and in chemistry there is always so many requirements for developing new medicine, new drug, or new process, or sustainable process, or green process, right? So there is a problem. You have a requirement for industry, we have a requirement for society. So there is a problem. Similarly in botany, similarly in zoology, similarly in life sciences, different. Even in computer sciences, there are problems, right? Now when you identify a problem, then you try to look for the literature. Now, very important word is literature. Nowadays, students are not doing the literature enough. So they are not aware that actually what is the problem in their field, right? So when you do the literature, you will find out that there are problems and there are solutions already being published. That solution already given. Now you analyze the solution, you analyze the publication, you analyze the published paper and books and different journals, and you review these publications and try to find out what is the scope, right? Scope means the gap of research which need to be plugged in, right? So the gap is very important. Somebody has already done some research. Now what? What we can improve? What we can change? What we can give better solution? What kind of solution we can give it? So that is called scope. And third one, and I mean fourth one is, now you put up a hypothesis. That is called rational hypothesis. That this is the existing solution right now in the available literature. Now I want to do this, right? So there is an existing literature and there is a gap and I want to do this. For example, right now we find that in the literature there is a drug. For example, there is a molecule which has some side effect, right? So there is a drug and there is a side effect. So you want to improve the drug so that there will be less side effect or no side effect. Okay, so there is a existing knowledge, there is a drug. Now there is a scope that you can improve it. So you put out a new solution, new improvement in the existing solution. That is called hypothesis. For example, cancer is a big problem right now in our society, right? So what is problem? Problem is a disease which is rapidly happening in our society, and we have no solution so far. So cancer, the problem. Now, how to treat the cancer? What is cancer? Different types of cancers, different types of solutions, medicine available. What are the side effects, the benefits, the lifespan? All we have to think, and that is available in the literature. Now we have to analyze the literature, and we have to try and find out the gap, that how we can get better drug. For example, if you take drug in the market which is targeted for cancer, the molecule, they are not specific, right? Or sometimes you say they are less effective. So how we can make it more effective? So there is a scope for improvement. Similarly, in every field you see, you will have a scope. You have to identify the scope. Remember that if there is a no scope in your research, you can't put up hypothesis. And your hypothesis must be validated by the results also. So why is very important? So if you can understand why I want to do this, for that you have to analyze the literature, you have to review the literature, right? Student, are you getting my point?
Yes, sir.
Okay, please.
Yes, sir.
Now, is this visible? Second slide is methodology. How? Are you getting this slide?
Yes, sir.
Yes, sir.
Okay, thank you. Now, this is very important part. Sometimes you put up a hypothesis, you want to solve the problem, you find out the gap, but you have to find the appropriate methodology to solve the problem, right? For that, you have to have an experimental design in your hand. You have a hypothesis that I want to make this medicine, which will be more effective and less toxic, for example. Then you have to make the appropriate methodology. Now, methodology can be partly reported in the literature or can be not reported in the literature. If sometimes some methodologies is already reported, you have to find out how can I improve this methodology also. New methodology, improved methodology, better methodology. For example, in chemistry field, if you are working in chemistry, if some people has developed through one catalyst, one process, you can use another catalyst for the same process or different process. So that means the methodology is known, but you change it for improvement, advancement, which of course should be more advantageous. Otherwise, there will be no improvement. Similarly, in lab sciences, in the scientific research of all the area, if you want to have a methodology improvement, first you have to look the scope where I can improve the methodology, or what is methodology already published. You must know it, right? So you have to do the experimental design. Again, you have to do the literature review. Now you see here the literature review is different than earlier slide what I am talking. Earlier you were looking for why, I mean hypothesis, a problem. Now you are looking for literature for methodology. How can I do this? What kind of methodology I can use it, right? So this is the another type of literature review you have to do, right? And once you finalize the methodology, you have to execute that methodology in the laboratory. And again, you have to analyze the result, whether methodology you have applied, which is working or not, which is giving you the expected result or unexpected results, right? So once you find out the methodology, you have to execute it, you have to apply it, you have to create your hypothesis in the laboratory is a realization through this methodology. So methodology is another part where you have to give the answer of how. Now, third part which I'm going to talk is the result and conclusion. This is also important, which is called the answer of what, right? So why, how, and what. Now you have a hypothesis, you do the methodology to solve the problem, and now you have to analyze the result, that this is my hypothesis, this is the methodology I have applied it, but now what is the result? Remember that your result must be in line with the hypothesis, then and then your research is in correct direction. For example, if you are working in a cancer and you're trying to develop a new molecule, new drug in cancer, and you find out that whatever hypothesis you have applied, and you find that your molecule is not working in the particular disease. Instead of that, it is working for some others. It's always possibility. That means your hypothesis is not in line with the results, right? You are getting some other results, right? That means either your hypothesis is not correct or your methodology is not correct, right? So your result must be in line with the hypothesis so that you can say that, yes, my research is in correct direction. Number one, you have to find out the rationale of your research. As I said that if your why, what, and how, if they are in line, that means your research is rational. Whatever research work you are doing, it is correct. It is in the same line, same direction. Sometimes it happens that you are doing the research and you are finding the unexpected outcome. And in fact, you will see the major discovery happening in the science, they are happening due to the unexpected outcome. You know the famous story of Alexander Fleming, right? He was doing some other research and finally find out the penicillin, which is the wonderful antibiotic, right? So it is always happening that you are trying to do some research and you get the unexpected result. That is called serendipity, right? So serendipity is also another way to find out new field of the research. So this new field of the research you get from the unexpected result. You have to again think about why, what, and how. How I can improve it, how I can do better, how I can do another improvement, like that. So you can develop a new area, new field of the research in your particular field of area or interest. Ultimately, I'm just telling that whatever research you are doing, it should be published. remember this word, that whatever research you are doing, it must publish. Rather than should, I'm saying that must publish. When you published your research, then and then people will know that what kind of work you have done. Sometimes what is happening is most of the research remain in the thesis itself. You are not publishing. Either student don't have time or guide don't have time to publish the results. So there is no use of this kind of research which is always remain in the thesis. Your every chapter, your every finding, your every units or whatever you give the name, right? It must be published. Suppose you are doing 5-unit thesis, your every unit should have publication, research publication. Because why I'm saying, when you publish your research, you have to give the answer of why, what, and how when you write the paper, right? Then you will realize that, yes, this is my hypothesis and this is the method I utilized and this is what result I have got, right? So that will give the beautiful combination and that will realize is a research paper. If you don't publish a research paper, you just write a chapter in your thesis and it will always remain there. It has no use because it is not going to benefit to the existing knowledge. If you want to add your benefits, I mean your part of research you want to add in the society, you must publish your research paper. Fine. Now, always I have a question. I ask the question that how we are going to find the answer of why, what, and how, right? So first of all, you need to do the literature search, literature review. Now, how to do the literature review? Okay, how can I get the more information that what is happening in my field? See, right now you are beginners, you are just starting. I would request all the students, please listen to me carefully. I would request all the students that you have a two choice. Number one, you go in the field where your guide is working, your research supervising is working. And number two, you create your new field, new area of research. There is a two opportunity. See, if the supervisor or if your guide allow, okay, you can follow their research field, your senior research field, and you can find out more additional benefits, knowledge, and you can find out new research paper and so and so. So that means you are utilizing your established methodology, established protocol of your seniors or your supervisors, chemistry or physics or biology or whatever they are working. Another trend is you try to find out what is happening in the modern research trend, right? How can I get that? So first of all, I'm hopeful that you know this word, research publisher. We call them as a publication house. This publication house, I am hopeful you heard this word. Are you aware about this? What is publication house? Anyone can tell me? Hello? Students, anybody know what is publication house? Have you heard this word? Publication, publisher.
Yes, sir.
Okay, good. So anybody can tell me the name of publication house, one or two?
Elsevier, Nature, Springer.
Yeah, great. Yes. So what is publication house? Basically, publication house, the publishers, basically they are publishers. For example, if I talk into the layman language, if you look at our daily newspapers, right? They are either Sandesh or Gujarat Samachar or Akila, whatever you think about. They are publishing newspapers. Also, they are publishing some additional publication materials every Wednesday, every Sunday, and etc. Right. So this publication house, they are publishing the informations. Rather, I would say they are publishing the research information, right? Number one. How they are publishing? They are publishing research journals. Remember, this is journals, not general. Every time you ask the student, what is research journal? They will say research journal. Journal is not the correct word. Journals. These research journals have a specific subject. Every research publisher have a list of journals, different field, maybe chemistry, life science, or whatsoever. They have a field, right? So they have different journals, subject wise. Even in chemistry, there is a more specific journals of inorganic, organic, physical, analytical, and so on. Similarly, in life science, botany, zoology, specific to zoology, and so and so. There are numerous. There is no enough number to say here. So they are publishing research journals. Now, what is research journals? How they are publishing? These research journals available in volumes and issues. Okay, in volumes and issues, they are available as a research paper. Now you see the line. So you have to find out the research paper ultimately. To find the research paper, you have to find out the volume number and issue number in the research journals. To find out the volume and issue, you have to go to the journal. Now to go to the journal, you have to find out the research publisher. Like one student said here that Nature, Science Direct, right? Bentham, and so on, Theme, and there are a number of publications. I am just giving some example next. So you first go for the publisher, find out the journal of your subject, then find out the volumes and issues, the past issues since the journal has started, and then you go for the research paper. Okay, so first, whenever you want to find out your research trends, modern trends, informations, you must go up to the research paper. Now, what is research paper? So this is the publisher. I told you that American Chemical Society, called SAS, Bentham Science, Science Direct, Team Villa International. There is the Indian Society also. There is every country has their society of either a different domain of the science, maybe chemistry or life science or biology, and so and so forth. Now, this research publisher, hello, please somebody put off your mic. Hello.
Sir, I have muted him. Please continue. Sorry for the inconvenience.
Okay. So these research publishers are publishing journal. Here I have given one example of chemistry journal, which is Bioorganic and Medicinal Chemistry. Now, just those students who have not yet searched too much or they are not aware about the literature, I'm sure that many of the students, they are not knowing that what is the journal and how we can find it, right? Because we are just beginning. So this is the name of the journal, and here you can see the title, Discovery of Novel Series and so and so. So this is the research paper title. At the bottom, there is a name of the authors who have published this research paper, and there is a short graphical abstract. So whenever you go to the journal, you will find the paper title, the author, and abstract and graphical abstract for particular journal, right? So you must understand that when you go to the journal and you find the volume number or issue number and you click on the title, you will get this kind of information as a research article or research paper. Now, another example of this journal, this is published by SAS, American Chemical Society. You can see here it is written SAS publication. The journal is OPRD, Organic Process Research and Development. Specifically in chemistry, if you want to work in the future as a in company or in industry, in R&D, you must visit this journal every day. See, I'm telling one of the wonderful techniques or tips I'm giving you, that if you really want to do the good research in your field, you must visit the journal which is in your field, in your research, which are published in good journals. You must visit every day. And you have to see every day what kind of research they are publishing. Also, you have to do the analysis of what kind of research already published, the different research paper, right, in that journal. If that journal is 50 years old or 40 years old, doesn't matter. Okay, you try to go through whole publications and try to see what kind of chemistry or what kind of biology or geology or whatever field they are published. Okay, so it's important that every day you don't miss it. Okay, like you are checking WhatsApp and Facebook every day, you have to check your journal every day. Okay, so when you go to the journal, you have to find out the research paper. As I told you, ultimately you have to have research paper. Now, how many types of research paper are there? Is this slide visible, students?
Yes, sir.
Types of research
Yes,
paper. So, if somebody is asking that you have to do the literature, that means what you have to do. Okay, if you want to do the literature, you have to find good research article in your field. Okay, now these research papers are basically 5 types. Number 1 is called notes. Number 2, I will show you also some example. End of the talk, I will show you how notes looks like, how communication looks like, what is the difference between notes and communication, what is the difference between letters and full articles, and so and so. But here just I am going to explain you that there are basically 5 types. Number 1 is notes, number 2 is communications, number 2 is letters, then full articles and review articles. Now, what is the difference between these 4? Generally, notes and communications and letters, first 3, are short research papers. Short means 3 or 4 pages. Generally, letters and communications are usually not more than 4 pages. Very short. But of course, very important. Remember here that there are several journals which are publishing only letters and communications. They are not publishing the full articles. What is the benefit? The benefit is that sometimes you are working in your field in research and you find out a good solution. For example, in chemistry, if I'm working in one reaction, okay, and I find out a new catalyst which can do the same reaction, but a better yield, better condition. Now, the reaction is known, but catalyst is new, right? For example, I'm saying this. So what I can do, I can publish my research as letters or as a communications, because the research is significant, importance, but it is not the full research, full length of the research, right? So I can publish that kind of research in communications and letters. Number 4, which is full article. Now, what is full articles? When you do the research in full depth and breadth of your field, for example, if you are doing the interdisciplinary research, which is connecting chemistry with biology, which is connecting biology with some other cells or biotechnology or nanotechnology, which is connecting to the another methodology or interdisciplinary research, you have a lot of explanation, you have a lot of data, you have a lot of hypothesis and the solutions and the results. Okay, so that kind of interdisciplinary research or the disciplinary research with full explanation, you can publish as a full articles. Now, the last is very important, review articles. Friends, remember that if you want to do a literature survey, you must be good in finding review article in your field. Remember my word, because review article is what? Review article means it is the review of literature in particular topic. Okay, suppose somebody has done research on corona since 2018 or 19, for example, 19, let's say. Okay, and if you want to know that how much research has been done so far in terms of the virus, in terms of the medicine, in terms of the spread, in terms of the mutation, etc. Right? So you have to find out a good review article published in that field, in that topic. So once you find a review article in that topic, you will get all the informations of your field, in that topic, up to certain years, maybe 5 years, 10 years, 15 years, right? Your depending on your field or depending on your subject, you can choose the review article length. For example, if you want to do the 10 years review article, if you want to do the 20 years review article, different review articles you can find out, and you will get the exact information that what kind of work has been done in this field or in this topic. Right? I will show you also that. So that is called review article. So when you want to do the literature, you must find out actually good review article in your subject. Then you can understand very well how many research paper already published. Most of the problem with student right now is that they don't know that how much work has been done and how much left. Right? It is always there. See, if you ask any student that, they know that what kind of work I want to do, but they don't know how much work has been done already. Sometimes what happens is they are doing the work in the laboratory, and after 6 months they find out that, oh, somebody has already published my work. Right? This is a common question always arises. So doing the literature is very important. For that, you have to find out the review article in your paper is very important. Until and unless you don't find the last stop where your field is now there, you cannot find out the gap. There are other ways also. You may have a question that, is this only the way we can do the literature? No. There are other ways also to do the literature where you can exactly find that whether your work is published or not. Okay, that I am going to tell you. But right now, if you find out the research article, communications, full article, letters, review articles in your field, and that's how you can create a literature. I'm just telling you from my experience that every PhD student should have at least 500 research article. I'm not telling the small number, I'm telling the medium number. It should be more than that, but at least you should have 500 literature review article in your field as a PDF file. Then you have to start your research. Why I'm saying this? Because if you don't do the literature enough, you will always find it a problem. You start your work, and after a few days or a few months, somebody will give you the paper that your work is published. Okay, so please make sure that you have done enough literature before you start your topic of research. Now, once you have a research paper, how to read the research paper? Is this slide is visible to you, friends?
Yes, sir.
Yes, sir.
Or you are getting my point?
Yes, sir.
Okay. So whenever you want to read a research paper, this is the 11 point that already there in research paper. I will show you the research paper end of the talk also. So it should have title, author affiliations, abstract, keywords, introduction part, which has figures and schemes and structures and photographs and whatsoever, tables, etc. Result and discussion part, conclusion part, acknowledgement, experimental, and references. In any research paper, whether it is a communication or letters or a full article, you should find this at least 11 part. Okay. When you write your research paper, again, when you write your research paper, you also have to prepare this 11 part. Otherwise, your research paper is not complete. Okay. So when you start writing your research paper, you have to have all these 11 parts. Fine. Now, these title, author, affiliation, abstract, keywords, you should be aware that what kind of information there in abstract, what kind of information I should give in author, author listing, first author, corresponding author, right? Co-author, everything you need to understand. So you start reading the research paper, then you will realize that how I can read the research paper and what part giving me what. See, if you see the part number 6, which is introduction. Introduction is always the answer of why, right? Why you want to do the research. That answer you will find in the introduction. Now, result and discussion, conclusion, that is what? That is the answer of what, right? And part number 11, that is experimental, that is methodology, 10, experimental, that is the methodology part. So why, what, and how, all these three you have to put in your research paper, or you can read from the research paper. Fine. Now, when you want to do the literature survey, I told you that research article is important. Another important thing is related reviews. So you first try to find out the list of journals, or you can say your choice of journals or preferred journals in your field, right? So first you find out what kind of work you want to do, why, right? Now you find out the journal which is publishing your work, your type of work. So you make a list of journals and you visit the journals, you try to find out the review articles, the research articles, and store it and read it. Okay. First, to do the research, it is important that you try to understand the research paper. You try to find out how to get the research paper. That is the important thing, right? Without that, you are not going to start anything. Then another good thing is books. And another one very good thing I can assure you, that is a SciFinder. SciFinder is a search engine. For example, in chemistry and life science, specifically, if you want to do the literature and you want to know that whether this has been published or not, remember that in your research, you have to do the unpublished work. You have to do new work. Okay. That means whatever work you are doing should not be published anywhere. Even not a part. Okay. Your entire work should be new. Okay. Maybe method or maybe all the work you whatever doing. Right. So whatever work you are doing is already published or not, right? You can find from SciFinder. SciFinder is a search engine, but I can tell you that this is not a free search engine. It takes a lot of money, of course, right? So maybe people don't have, specifically in academia or university, we may not have SciFinder access. In some university may have, but in industry, most of the larger industry or bigger industry, they have a SciFinder access. So you can take the help of your friends or seniors and colleagues and your professor or supervisor, whoever you're talking. You can try to find out that your work is published or not from SciFinder. Similar search engine is Reaxys. Reaxys is also the similar search engine which can give you whether your research work has been published or not. In these search engines, you can search by title, you can search by topic, you can search by structure, you can search by author, you can search a lot of things, and you can find out the latest work whether has been published or not. Then PubMed and Scopus, and for patent we have USPTO, also have Indian Patent Office. So you can try to search the Western region patents or US patents or Indian patent office. So they are also the part of the literature if you want to search the patents. Okay. So literature survey is not only research article, it is also from, you can do from the other search engine like SciFinder and Reaxis and all this kind of search engine available. Are you getting this, students?
Yes, sir.
Fine. Now, when you do the literature survey and you done the research, now you want to publish your manuscript. Okay. Still, I have not started the research proposal. Still, I am talking about research paper only. So you go to the journal first. You have a manuscript in your hand. Manuscript should be in usually in Word file. You prepare a manuscript. Manuscript preparation requires a lot of time. You have done the research in the laboratory. See, I'm telling a clear truth that if you are a good researcher in laboratory is not enough. You should be equally enough for writing. Because if you have good results in your laboratory, in your hand research result, I'm talking, and if you are not good writer, your research will not published in a good journal. Okay. So if you have good research data and you want to publish it, you should be a good writer as well. It is like a 50-50. Okay. You should be all-rounder. You must know the bowling as well as batting. Similarly, as an all-rounder, you must do the laboratory work. Equally important is writing manuscript. Okay, so you must have good command in the language, specifically scientific language. I'm not talking about English. I'm talking about the scientific language. How chemistry, how life science, how biology, how geology, or whatsoever topic is there, or research field is there, you must understand how to write in the manuscript. Okay, so when you want to write the manuscript, you have to first choose a journal. For which journal or in which journal you want to publish. You get the author information from the journal. You have to choose whether you want to publish as a full article or whether you want to publish as a letter or whether you want to publish as a notes or communications, etc. Okay. Then you prepare manuscript according to the journal guideline. The journal will give you the clear guideline that how you are going to prepare your manuscript, what kind of Word file you are going to use, which type of font you are going to use, what will be the margin, what kind of figure, the size of the figure, the size of the table, the size of the photographs, everything. They will give you clear instruction. So you prepare your manuscript according to your chosen journal, and then you prepare a cover letter. This is also important. Here, the cover letter describes the short story of your finding, of your research, and you address it to the journal editor. Also, you include the reviewer list also. Now, what is reviewers? The reviewer means every time journal will ask you the name of 3 reviewers which are relating to your field. Suppose you are doing work in X field, there are some people already working in that field, I mean professor or researcher or whatever. You must know their names, that who are working there. Okay. And you have to give the name of reviewers to them. Then you prepare the graphical abstract, right? Now you submit the manuscript, cover letter, graphical abstract, and online submission. Nowadays, no hard copy submission. Everything you have to submit online, right? Then you will get a response from the editor of the journal. I'm just giving very shortly this story, otherwise it will take a lot of time. So I'm just giving you the short highlight how your manuscript will travel. Okay, so you will get a response from the reviewers, and there will be 3 possibilities that your manuscript is accepted, or it needs revision, or if it is rejected. I can tell you that in good journal, the acceptance chances is about 10 to 20%. 70 to 80% either revision or rejected. If your paper is rejected in particular journal, don't be afraid or don't be frustrated. You have to choose the another journal, okay, which is also similar type of journal which can publish your research work. So there is hundreds of journal, okay. Even if I tell you the figure, more than thousands are journals in there, more than 1,400 journal only in Scopus index. So there are thousands of journals are there. So no need to worry. But you must prepare your manuscript. As I told you earlier, that you must have good writing skills so that it can be published in good journal. Okay. And once your manuscript is accepted, your contribution is acknowledged in the field of particular research, right? And you will have a great satisfaction and great joy that yes, you have done something new in this field, right? So you must understand this sequence. Fine. Now, so this is the journey so far, which I am talking about the manuscript. So we started from what is research, hypothesis, methodology, and why, what, and how. Three part. Then how to do the literature survey, how to read the research paper, what kind of different parts are there, and how to publish the research paper. Of course, there will be other professor, they will explain you in detail that how you are going to publish research paper, but just I give you the highlight. Okay. So now let us go further and try to understand how to write a research proposal. Is this slide visible, students?
Yes, sir.
Fine. Research proposal writing is important because when you want to write for Sodh scholarship, or whether you want to write for Inspire fellowship, or whether you want to write for funding, especially to get the money from different agencies, right? We need to understand how we can write a research proposal, because in the future you also require this, right? So generally the research proposal we are writing to funding agency, right? The purpose of writing research proposal is to get the fund from different funding agencies. In India, it is UGC, of course, DST, Department of Science and Technology, SERB, Science and Engineering Research Board, and GUJCOST, which is especially in Gujarat, right? So you have to target your funding agency, whether you want to apply in one of these funding agencies, right, as a research proposal. Now, how to write a research proposal? What kind of different parts we have? This is one slide I am trying to explain you that every research proposal has this part. These 7 parts, 7 criteria you can say. Number 1 is the title of research proposal, right? You have to give a title of your research. Number 2, origin of the proposal. That means why. I told you why you want to do the research, right? What is the problem in the society? So it is called introduction of research problem. Now you have to put the review of status of research and development in the subject. That means you have a hypothesis, you identify a problem, you find out a scope. Now you have to find out how much work already has been done. What is the status in this field? Okay. Remember that right now, remember that you are not the first one to discover something. There is always some reference. There is always some status already there. So you have to review the status, that how much work has been done in this field, right? That's what I told you, the literature survey, right? So research and development in particular field, you have to put there. That is called review of status of research and development in the subject. Here also you have to introduce the literature review, you have to find out the current status and scope and significance of your research. Then very important part is objective of proposal. Why you want to do this? Different objectives. Then how you are going to do this? That is methodology. And how much financial assistance required. And of course, reference of your related research field. Remember here, you should not copy somebody else's work. You have to maintain the ethical practice. Okay. Plagiarism, we call them as a plagiarism. You should not do the copy and paste from other research papers or other research proposals. You have to create your own research paper and own research proposal. Okay. Of course, you can use their knowledge, you can use their words, you can use their sentences in a particular way that you can write it newly. You have to write in your own words. Anyway, so this is the different parts you have to do the research, how to write a research. Now let's try to understand that individual part. Now, title of research proposal. Number one, this title should be concise. It should be describe the objective as well as outcome of the proposal, and also you should not use the abbreviation, right? So title should be very short, it should be very informative, and should include objective as well as outcome of the proposal. Okay, so here I have given the title of some of the chemistry proposal, right? For example, design, synthesis, and bioactivity of quinoline derivative. So here, what I'm talking design, synthesis, and bioactivity. So my objective is clear and my outcome is also clear, right? So I must include a concise objective and outcome of the research proposal title. Some other example I already put here, you can refer it later on. Now, very important part is the second part, which is origin of the proposal. Once you decided the title, you have to find out the problem of your proposal. So here you have to give the little bit introduction of your research problem. What is the problem in your field? Then its social relevance, very important. As I told you that cancer is a problem. Now it has a direct social relevance, right? So you have to describe the problem and its social relevance, how much it is going to affect with the society. If you solve the problem, how much it is going to benefit with the society, right? Then your proposal should include the improvement in current condition or status, as I told you already. And also you must put the appropriate methodology. So there are several problems, like process chemistry is a problem, synthetic methodology is also a problem, medicinal chemistry, you can find problem in there, natural products, you can find problems, right? So there are a number of ways you can find the problems. Now, review of status of research and development in the subject. As I already told you that you have a title, you know what kind of field I'm going to choose. Now you have to do the review. For review, you have to prepare 4 category or 4 different parts. Introduction part of the literature survey, then what is the current status in that field, progress in the field, what is the scope, and what is the significance of your proposal. Okay, what is the importance of your proposal you have to showcase here. Now you have to write the objective. Remember that whenever you write the objective, you have to start generally with 2, TO, 2, to develop, to prepare, to extract, to do something, whatever, right? Important thing is your objective must cover your aim of the research study. For example, you want to do the research in cancer, and if you write your objective here, instead of cancer, something else, or general, which is not good. So it should cover your aim, research study. If your research study is larger, if your proposal includes the lot of multidisciplinary research, then you can prepare or you can write individual objective for individual plan or individual topic, right? Separate objective for multidisciplinary research, right? So, for example, you want to develop a new method, you want to develop a new heterocycle, you want to extract some isolate natural product, and so and so. So that is called objective of the proposal. Now, methodology. Methodology is what? The methods, the experimental design you want to utilize in your research proposal. So you have objective clear that I want to do this. Now how to do this? So you must apply the experimental detail, right, to showcase the problem solution. Suppose you have a 5 objective and you can put separate methodology for all objective also. Sometimes your objective can be mixed type. You can put 2 objectives together and the methodology also together. Right? Here you can provide the scheme and diagram. For example, if you are working in chemistry, if you are working in biology, if you want to showcase the flow chart or diagram or the signaling pathways or this kind of information you can provide in the methodology part. And also you have to cite the references. You should always do the referencing every time, which is known. Okay, if method is known, you must cite the reference that okay, this is known and this I want to do, right? So you can include these new methods, new design, and so on. So that is called methodology. Now important part is financial assistance. So to ask the money, it should be in separate head. You have to ask the financial support in separate head. This is the general, I'm just giving the general idea of different heads. There is one head called instruments, right? In your research proposal, you want that some instrument required. You want to purchase it, okay? So you have to give here how much money will require for, this is just explanatory, okay? How much money you require for that instrument, particular instrument, or more than one instrument, okay? Then there is another part is consumables. Consumable means which is going to utilize, like chemicals and glasswares and hiring services are called consumables. Consumable means it is going to utilize, okay? So you can figure out in your field what kind of laboratory equipments you will require. Then you can, there is a third part which is called the books, journals, or subscription of particular publications or computer softwares, something like that. You can use that part to ask some money. You can also ask to some money for travel within India. Of course, this is not a tour, okay? This is for to showcase your research, okay? To present your research. Right? For that you require travel money like a train ticket or so and so. Okay. And contingency. Contingency means the accidental expenses, which you have not thought of, but sometimes it happens that your instrument breaks off or sometimes your important assembly breaks up, something like that. Then you require some contingency money so that you can utilize it wherever it is required. So then you have to give the total, that this is the total. For example, here I put 6 lakhs, right? And also you have to give the year-wise plan. If your research project is 3 years, then you have to give every year plan. First year plan, second year plan, and third year plan, and what kind of target you are going to achieve. Okay, so that's how the financial assistance you can ask from the research proposal. And last, you have to give the references. Now, references, what is the references you must understand, right? So you can cite the reference of books and journals, right? Then very important thing is all the reference should be in same format. Sometimes what happens is students writing the reference very arbitrary. The pattern, the format is not unique, so it's not looks good. Okay, so you have to follow the pattern of writing referencing. For example, here I have given at the bottom here. So I start with the author. You see, this is the name is author first. Then once the author list is complete, now I'm starting with the title. The title is Synthesis Evaluation. That is the title of research article. And once the title finishes, I have bold out the name of the journal. That is Journal of Medicinal Chemistry. Then 59 is the volume number. 2016 is the publication year, and 5264 to 5283, that is a page number of this research article. That means if I go to this journal and if I try to find out the volume number 59 published in the 2016 and find out this page number, I will find this paper. In this paper, they have described the synthesis and evaluation and mechanism study of novel risk compound for anti-tumor activity, right? So that's how referencing has to be done. And that's how you have to write every time in the same manner. Now here, one thing is very important is you should always give the reference whichever is available. Do not undercite the reference or research already published. What happens is students, if they are working in their field and they find an article which is close to their work, they will not give the referencing list or they will not include that in referencing list. That is called undercite. Of course, you should not undercite any reference, no matter how close your research to the published research. Of course, there should be difference, little bit difference, no problem. If there is a little bit difference, no problem. You must cite the reference which is published already there, which is very close to your research field. Okay, so that is called under citation. Don't do the under citation. You must give credit to people who have already done some research in that field. Okay, so that is called very important referencing part. So all this part I try to explain you how to write a research proposal and how to do the referencing. So with this, I want to complete my talk of how to write an effective research paper. Now, before I close, I just want to show you the different types of research paper. So for that, I will stop this and I will start new slide. Student, are you getting my points or not?
Yes, sir.
Yes, sir.
Okay. So now I'm trying to present the screen. My hopefully it's visible to you. Is it visible?
Yes, sir.
Okay. So first I will show you this research paper which is published in the chemistry. Are you getting this JOC note? Is it visualized?
Yes, sir.
Okay. So friends, here look at this paper. Here it is published in a journal which is called Journal of Organic Chemistry, JOC. And here it is clearly written as a note, NOTE, means this is a research paper but published as a note. Okay. Here there is a title, here there is an author. Everything is there which is already known for other research paper. Okay. But here the difference is, usually I told you that notes are smaller research article. For example, this one is only 4 pages. 1, 2, 3, and 4. Just 4 pages, right? So this is called notes, smaller research article. Of course, it is important. Remember that if it is only 4 pages, that doesn't matter that this is not useful. No, this is very useful. But the results here published as a notes. This is not a broad targeted research, but a smaller topic research. Now let us see the communication. How communication look like. Is this visible, friends?
Yes, sir.
Yes, sir.
So here you can see this research paper. Here already written on the top, that is short communication, right? That means this paper is not a full article, it is a short research article, and also it is 5-page only. Okay, so this is also a published article. Here you have title, you have author, you have keyword. I told you there are 11 points in any research article. You will find everything here. In every research paper, it doesn't matter whether it's article or whether it's short communication, doesn't matter. You will find everything. So introduction is there, abstract is here, right? So result discussion part is there, right? You have tables and figures and schemes are there. Conclusion is there. Experimental section is there. Acknowledgement section is there, and reference section is here. So everything is there, right? So this is called short communication, but only 4 to 5 page, maximum 5 page. Here it is about 5 page. Now let us see the another, we call them as letters. This is the letters. Is it visible, friends, from Science Direct?
Yes, sir.
Fine, thank you. So this is published in the tetrahedral letter. This I published my first rearrangement. I told you in the beginning. It is a new rearrangement found in the field of chemistry. So this is published as letters. Now you see here, the title is there, author is there, institute is there, abstract there, introduction part is there, scheme, result and discussion, right? You will give the data to their informations, conclusion, experimental part, acknowledgement part. Sometimes you can give the supplementary data which is useful for the readers, and referencing part. So here again it is only in 5 pages. Total page is 5, but it is called as a letters. So communications and letters are usually 4 to 5 pages. Right? Now you look at the full articles. Now I'm showing you the full articles. Is this visible, students?
Yes, sir.
Fine. So this is the publication I'm just showing. Is a Bioorganic and Medicinal Chemistry journal, which is published by Elsevier, Science Direct. So here also, now you see this result. Okay. Now this paper article, this research article, you will see the page is 12 page now, from 1264 to 1275. In any journal, any research paper, if you look at this here, this will first give you the name of the journal, volume number, publication year, and page number of this research paper. Okay, so you have to read like that. So we have a title, this is the author, then abstract. Now here just for a few minutes, for 1 minute, I just want to tell you the article history on the left hand side. This is important. Okay, just try to showing you. Can you see the article history?
Yes, sir.
Yeah, right. So you can see here, received 22nd October 2008. That means I have submitted this paper on 22nd October 2008 to the journal. Then it goes to the reviewer, 3 or 4 reviewer, which is working in your field, and they will read your paper, they will understand your paper, and they will say whether your research paper is publishable or not, whether there is a problem with this research paper or not. Okay. If there is a requirement, they can revise it. So you can see there is a revised on 8th December. That means it was submitted on October. After almost 2 months, it was revised and it was accepted in December. Okay. So you can understand that any research paper require at least 2 months. This is the minimum requirement. At least 2 months to publish from the date of submission. You submit today, you have to wait at least 2 months. If you are fortunate enough and your reviewer say yes, then you can publish. If your reviewer says no, you have to find another journal, and again you have to go for the review, and again wait for the years, and then you can publish it. Okay. So this is a continuous process. So that's, I show you that every article has the history written, that what are the different dates included with this paper. So abstract, then introduction part, then chemistry part, I mean the experimental biological discussion, different schemes, the different data I have given, the experimental in vitro, in vivo results, then very experimental detail. So here, friends, you can easily distinguish the two papers. Earlier, I show you the letter, and here I am showing the full article. Right? You can understand that the full article is having more data and more depth research. Right? It has a biology, it has a chemistry, it has a in vivo study, animal model, everything is there. Right? So that's why it is called the full article. Okay. So reference and notes. Now, when you write the reference, you can see that here every reference has the same style. You can see the author name and then journal and the publication year in bold and then volume number and then page number. So it is a unique style for every journal. Okay. So this is about 12 pages. So this is called full research article. Now you see the review articles. I'm hopeful you will be understanding. Is this visible to you, friends? Review article?
Yes, sir.
Now you see the review article. This is a review article on one reaction called Biginelli reaction in chemistry. Okay. So look at the title, past, present, and future of Biginelli reaction. That means what? This author had tried to put up all the data of one reaction in this review article. Okay. This review article is of 68 pages. This review article has 68 pages. Okay. But remember that this is a compilation of data. It's kind of a compilation, aggregation of data. So, which is already published in the different journal, different research paper. But every review article should be having a specific title. For example, this title is only for Biginelli reaction, one reaction only. Similarly, you can have a different title of your interested topic. So, this is a 68-page review. Now you can imagine that if you have this kind of good review article in particular your field, then you can get all the informations, the latest trends, the latest research, what has been done in your field from past to present, right? So you must find a good review article in your field. Remember that review article is also research article, right? Review article are also called research article because whatever they are writing or they are gathering the data, it has not been done earlier. Okay, it has been not published anywhere. That's why it also publishable. So this is called the review article. If you look at this, I'm just directly going to the end of this. So see here, this review article has cited 542 references, right? That means enormous work they have done to find out the research paper and compile them and aggregate them in a particular way so that they can write this review article, right? So you can also write a good review article, because to write a review, you don't need to go into the laboratory, right? You have to do it on computer. You require a good literature skill. If you can do a good literature review, you can write as a review in this field. Okay.
Dear students, we will start next sessions sharp 2 p.m. Kindly present at 2 p.m. Thank you.